Respected chairpersons, respected seniors in the audience and colleagues. First of all, I'd like to thank the PeSuCON team, Dr. Ramesh and Dr. Deepthi for giving me this topic. So Dr. Ramesh called and said, talk about hypospadias, changing trends. And I thought, good, that's not a bad thing. And then I looked up the topic, it was recent advances in hypospadias. So that was a bit of a bouncer for me, so I'm not sure whether I do anything recent. So I went and looked up the literature about what it is. I think when we do a talk, it's not only us trying to say something, it's us learning, and that is what is good about doing a talk. So what do I know? Is there anything new on the horizon about hypospadias? We've all read about hypospadias, we've read our textbooks and stuff. And so I had a search. I looked at the last 3-4 years. And for me, what I think of a topic like this is to bring out what may be new. And I'm not going to be talking about operations because, as you know, all new operations in hypospadias have to stand the test of time. So we'll talk about what I think was interesting. So looking at different things in hypospadias, we'll look at how things started off, the etiology, what are the genetics involved, what is the management, and what are the new things. So we'll try to go in a structured manner. So I looked at this, and this is a study from 2016 actually, and it looks at increasing hypospadias in USA. So they've actually showed a prevalence which has increased from 6.1 to 6.8, which doesn't seem a lot, but their numbers are in millions. So this is 1.9 million, 1,917,000, that's right. So they've actually shown that there's been a slightly increasing trend in the US, especially on patients with small gestational age for birth, multiple gestational age, premature births, and at least 20% of these increases have one of these risk factors. So definitely we might be seeing similar things because we also have this particular group which are now going more and more in India. So there is an increasing trend prevalence for hypospadias. We know the etiology, prematurity, issues related to birth weight, what I just said, mother's age, exposure to endocrine disruptors, genetic. Are there any other things that, when I read was not very clear, or is there anything new that is coming in about etiology? And I saw a few articles. So this is one from South America. So they looked at 4 million newborns across 20 centers. And they actually did an analysis. Again, this could be a lot of statistics. So they've gone into detail about the discussion. But they looked at altitude above 2,000 meters, and they show there is less incidence of hypospadias with an odds ratio of 0.59. So if you look at their numbers, this is again a slight anomaly which they got, which they couldn't really explain. But the numbers, which was above less than 2,000 meters, is going slightly up or the same, and the ones below above 2,000 meters is dropping. So again, they looked at clusters and stuff, and they could not actually explain. So if you live very high up, probably you won't get hypospadias, is my inference. Then the other opposite, maybe it's colder there. So a Turkey study looked at higher ambient temperature during pregnancy. We know that high temperatures in the 70s, they've done studies, 60s and 70s, do cause anomalies. But they did a 15-year retrospective study, so you have to take this with a bit of, you have to take this carefully. They did have a large number of patients, about 5,000. And they've also mentioned that, there's increased incidences in warmer countries like Spain, South Italy, Australia, Puerto Rico, African countries, and probably a little lower in Norway and Japan. So in their study, they had a higher temperature of 36.3 degrees, and they showed an odds ratio of 1.32. So they postulated that it could be heat stress to the mother during this time, which had an hormonal influence on the baby and could cause a higher chance. So again, family history. Now, when we talk about hypospadias, we do take a brief family history, but not much. So a group of French team went back and looked at the histories and families in a prospective manner, and they looked at about 395 boys. And they got a figure about 22% of these patients actually had a family history, which is a lot more than I would expect. I would be looking about 5% or maybe 10%. So they actually went back and did questionnaires, called them back. They had to do 2 or 3 interviews to find out. And they said if you do look closely, that there will be a relatively high family history. And father, among these 30% was the father. So this bit we'll get easily. But the remaining, if you look at it, the family history is probably much higher than we would think. So we do know that the familial cases also have a higher genetic defect. So they have a higher incidence. And this goes across all types of hypospadias, from the proximal to the distal as well. So the family history is a lot more common than we would think maybe, let's say, 20 years ago. So one of the editorials in Journal of Urology in 2018, he says that hypospadias, is it nature or nurture? So we know that genes are involved. The study that I just showed you in that same article in the Journal of Urology 2018 showed that familial cases are more common. We know that identical twins who have the same set of genes, one can have hypospadias, one may not. So we can't say it's just gene. And then we know that environmental factors also cause hypospadias. But what we know nowadays about environment factors is that they do cause epigenetic modifications, which for me in a layman's term means that even though the gene is the same, how it actually is converted or translated or transcribed, or how it is passed on changes because of the environmental factor. And the interesting thing about this is once that gene is affected or the transcription or translation is affected, this can be passed on to the child. So you can get a higher history of familial or so-called genetic issues with environmental factors. And he goes on to say that, this can have implications for fertility, and maybe we can prevent it by modification of lifestyle, maybe not being exposed to a high temperature or whatever causative factors, and it might have to be involved in genetic counseling. So if things can cause epigenetic or genetic issues, environmental factors to cause hypospadias, maybe in the future we'll find something that prevents the hypospadias occurring. So is it possible that treatment is possible if we find out more about epigenetic factors? So again, looking at molecular expression on mouse erythroplates in 2016. Now we have a few things called genetic sequencing and microarray, where instead of looking at one gene at a time, you can just put an array of genes and they can look at 100 genes at a time. So which this was not available 10, 15 years ago. So this particular group of people looked at a 20-fold increase in expression along the urethral plate. So any gene which is 20 times more than normal compared to the surrounding area, they consider that it could be involved in actual development there. So looking at that, it's nice to have colorful photos. So this is a nice colorful photo. The urethral plate is there, and they actually identify 84 genes in that area. Some of the genes are associated with molecular function, the green ones. Some of them are related to pathways, and some are related to biological processes. So they've now identified 84 genes which are related in some way to formation of the urethral plate. So this is from 5 or 6 or 7 before to now 84. And obviously research is continuing on this. So now let's come to antenatal diagnosis. So we have seen that it is being more and more common identified, and recent article in Journal of Pediatric Urology, they looked at 25 cases, prospectively they looked at 25 cases of antenatal suspected hypospadias, and they confirmed 18 of them. And another 3 had some other associated anomaly like an undescended testes or an abnormality. So they give us 72%. So the last 4 or 5 years I didn't find many articles around antenatal diagnosis, but I'm sure with our present increase, we actually can diagnose them better. So again, looking at this, this is a foreshortened penis. You can see here the curvature here. Again, the scrotal sacs next to the penis which seems short, and here again scrotum and the penis, and this is called the tulip sign. So it is identifiable, and probably with more and more accuracy in the future. So we will get an idea about it, and we'll have to start counseling parents regarding implications. Anatomy itself, is there any changes that we have thought about it? So in 2018, JPU again, Derlick, I think his name is, talked about that the epithelial surface that we see on the urethral plate is actually larger than we presume. So he says there's a lot of folding of this epithelial plate, and there are a lot of epithelial adhesions. And now we know that during development we have a canalization of the urethral plate, and then the open zipper, and then finally the closing of the zipper to form the urethral tube. So this is in the open zipper part of development. So they looked at 72 patients, and they talked about dissolution, or dissection is what I think they mean when they say dissolution, and it increased the urethral plate size from 5 millimeters to 7.8. So looking at this, this is what he talks about. If this is the urethral opening, sometimes you see a groove here or an opening here or a small kind of dimple here, and if you look on the side view, you can see that this actually extends further down. He takes, so this again the same thing, it's a groove there and there's a small area there. If you do a cross section, you can see that the epithelial plate is actually going down, and this is a histological representation of that. This is, you can see the epithelial lining here, and it actually goes down into the glands further. Okay, so this actually can be used as an epithelial lining for our tube, and he's got these from either patients who have died for certain reasons, and also he's taken the urethral plate distally when he's done 2-stage procedures. That's where he's got these. And going on, he shows some photographs. I think we've all seen some patients where you groove is very shallow, you just separate it a bit, it becomes deep. And so he's showing that that can occur. Again, looking at this, you can see the groove there, and he just opened that. He's dissected out. He's not cut anything, is what he says. He's called it dissolution. So you see, I presume that's dissection in his terminology. And you can see here that these grooves are actually much deeper. So they get an average of 2 or 3 millimeters, which would be very helpful for us to tubularize, obviously. So what else about the anatomy or the structure that we look at hypospadias? We know that they're not normal tissues. We all know that. So I think now that we're looking at what is called an electrosonographic or ultrasound evaluation of these particular hypospadic patients. So they use something called sonoelastography. We talked about splenic stiffness in the portal hypertension, similar thing that they're using here. So it's a special software, and they use ultrasound to determine the elasticity of the tissue and differences. So these group of researchers had 175 patients, post-op, pre-op, and normal non-hypospadic patients as well. So they looked at the pictures. If you see here, the red is soft, so that's the ultrasound, and this is nice and soft, and the blue is hard. So they had 3 gradings, and they found out that the corpora spongiosum was globally stiffer along the length, proximal to the hypospadias, and the corpora cavernosa was also less developed. And these changes extended to up to 15 millimeters proximal to that. So if you think about tubularizing these structures and they are stiffer and firmer than usual, then you can understand why some of your surgeries have actually failed, because the tissue is not actually normal there. So they're not normal, stretchy, pliable tissues. So they're looking at those. And then we look at biomechanical properties. So again, the Journal of Pediatric Urology in 2018, they had a look at the biomechanical properties of the urethra just at the opening and proximal to that. So they use a catheter called an endoflip, which is basically an impedance-based, which measures it by impedance, like your balloon catheters or urethral profile catheters, and it has a balloon inside. And they use pressures at 40 centimeters and 60 centimeters of water, and they looked at 19 hypospadias versus 7 control before surgery, pre-op before anesthesia. The control, I think, were all circumcision or other surgeries not related to hypospadias. Now looking at some patients, so if you look at this graph, what you look at is, here is where they've increased the pressure from 40 to 60. This is the actual millimeters stretch. So this is the urethral meatus. So when they look at this, they can see that at 40 there's a bit of stretching proximal to the hypospadias, and once they go to 60, the stretching is good. So these are both normal patients. This is when it's converted to 40 to 60, and that is in millimeters, 4 and 5 millimeters. And these are hypospadic patients. You can see that the change is not as much. They've done that in 3 dimensions as well. So this is the urethral opening, that's distal, this is proximal, and this is up to 5 millimeters, the red. So this is again at 40 centimeters of water. At 60, it increases in size. Similarly 40, here the opening opens up well, here not as much. These are normal, and these are hypospadic patients. In here you can see there's hardly any change at all, even under 60 millimeters. So that tissue is not at all pliable at all. So definitely there's some abnormality. So they thought the urethra itself was narrower overall and less viscoelastic. So obviously this will probably mean that it is more difficult to repair, and they'll have long-term issues with urinary flow after repair, possibly. This is the implication of this test is. So present status. So looking at this again, another review article in 2017 about current challenges with proximal hypospadias. The problem for us is now, 20, 30 years ago, our bosses used to do them one way. We do them slightly differently. In between, we had something in between. So to compare these, it is very, very difficult. So all the studies that we've done are variable. The methods they've done to analyze this and the methods of surgery they've done are variable, and there's no standardization. So it's very difficult to compare a hypospadias repair of 30 years ago to something that we do now. And the results, because they're so long, all the techniques were very different. And randomized control trials in hypospadias it's not a really good way to study hypospadias, and it's not something that should be done in hypospadias. So what he recommends is that we should do a standardized pre-op assessment. Now there are a lot of charts that we can get, and also standardized reporting. Obviously the reporting has been very variable. I think you see some people say their problems are only 1%, 2%, 5%, and some people say it's 40%, 50%. So it's very difficult to believe which one it is, and are people doing different surgery. So it's very, very difficult to compare. So they talk about something called SCAMP, which is Standardized Clinical Assessment and Management Plan, which is basically a group of people getting together, or centers, which analyze them in a similar way, pre-operatively, and manage them in a similar stepwise manner, and postoperatively another in a similar way. And then he brings out the thing is if for proximal or difficult hypospadias, should we have centers of excellence? Like we talk about biliary atresia for transplants, should we have centers which do that? So he goes on to say, if you look at these complications, and this is Snodgrass at the top and there's others in between, it varies from anywhere from 3 to 50, 68%. Okay, so you see a wide range, so 4 to 68%. So he goes on to say that time itself, on a commentary to that particular thing, is time the most important variable. If you wait longer, are you going to see more problems? So we know that 30% of at least 30% of them have some long-term recurrent problems. Now this is a real issue for us. When I was training here in India and after my training, we used to follow hypospadias for 2 years, 3 years. Then when I understood a bit more, I follow them till post-puberty, till 16, 17, after their pubertal growth spurt, because I know they can run into trouble. But we might have to do it for longer. So, and again an editorial, Hypospadias as a chronic illness, Journal of Urology 2018. So distal hypospadias complication rate is 10%, proximal. The longer the follow-up, the poorer the outcome. So most of the operations are done at mid-30s where the complications occur. They include lower urinary tract symptoms, urinary tract infections, and maybe stenosis or stricture of the repair. So European Journal of Urology 2017, they looked at redo surgery. So if you see these articles, they had an assessment of these, and most of them are in their 30s, again 39, 38, 37. And one group saw 54% erectile dysfunction. So I don't know how much we think about this. And another group said they were very difficult surgeries and multiple procedures may be required. The other group said the voiding problems were about 82%, UTI 36, curvature, and BXO in these patients, and fistula. And again, one more group said they had a 12% failure rate. So there are a lot of long-term issues that we are not maybe thinking of or warning the parents about. So in UK in 2017, they had a look at their results, 24,000 patients approximately, and looked at all patients above 20 cases they considered higher volume center, less than 20 as lower volume. And they looked at 75 centers in England. They thought the higher volume centers had less complications than the lower volume centers, 25% to 75%. So that is their finding. So again, I'll leave you to interpret what that means. Huh? So now coming to any surgical things that or other things that are really recent, tissue expanders, this is again not very recent. So they talk about tissue expanders. They put them dorsally under here, and then the tissue expander goes, dorsally, and the pump goes under the suprapubic area, and increase it every 2 weeks for 12 weeks. So this is a 17-year-old patient they've shown. They've shown nice tissue expansion and nice cover. And so this is not a new study. So there are quite a few studies, and I personally think that this is something that we can also think of for use for hypospadias in the future. But again, in children, probably not. As you see, this is a slightly older study because they talk about cripples. We don't use that term anymore. So let's look at other things as well. So in the adult world, they are doing tissue engineering for urethroplasties for strictures as long as 5 centimeters. So this is a study which they had a median age of 57 years and a median length of stricture of 5 centimeters. So they made a tissue engineered oral mucosal graft. They took oral mucosal biopsy and sent it for forming a tissue engineered graft and then put that into the patient. So they had 38 patients and an 84% success for a 5 centimeter length stricture. And I hear in Bangalore also some of the urologists, there's a few urologists who are now using tissue engineering for their adult strictures. So I think this is something that we might need to be thinking of in the future. So again, this is another study where he did it in rabbits, autologous granulation tissue tubes. So he used male rabbits, he put a silastic tube, and you know when you ever put your port or anything, you get a very nice cup around the port. So they made a granulation tissue or the cup which is formed, they used that and harvested and they put it back into the rabbits. And they found that it was quite useful for anything about centimeter length in the rabbits. Anything more than that, they had issues with vascularity. So half to 1 centimeter is good. So again, something that might be applied for us clinically as well is something to think of. Now let's go a bit more research-wise. So tissue engineering for severe hypertension, is this the future? So this is from the Frontiers in Pediatrics 2018, and they go on to say that it's 50 to 58% of proximal hypospadias do have complications, and there are different ways that you can tissue engineer. You can use acellular scaffolds, which is just a basic framework. You can add cells to them, a cell-seeded scaffold. You can tissue engineer, means you do it outside, and you can make constructs as well, not just a flat plate. So we know if we do this, we need them to be biocompatible. They need to vascularize. They need to degrade later on. And local tissues, the urothelial lining, the musculature needs to regenerate in that. And obviously it should be nice and compliant for urine to pass through. So there are things that can be used. There are biodegradable things. There are artificial things. You get natural silk fibroin. You also get CIS, which is now available, small intestinal submucosa. You can use cell-seeded scaffolds. Probably for longer defects, you need the cells because the vascularity is not good. You can use oral mucosa, or they used to use urethral cells. Some people used to do open the bladder and take it, but you can use it by getting invasive bladder washes you can use now to get the cells to see. Okay? So that's what they've been using in some of the animal models they're using. Or you can also fabricate, tissue engineer them. So you can make a scaffold like this, put some cells in it, rotate it down and put some more cells and make it into a tube. Okay? And cell seeding for longer strictures is essential, is what they think. So again, here is a photo of CIS. So that's the thing. And they're using CIS as a onlay here. It can be used onlay, inlay as well. And they're closing it over. So maybe that is useful. And this is something that can be pretty much done nowadays as well. So we can start using that if you get the CIS graft. So again, looking at research. So there have been patients done. 4 patients here, 8 patients here. And so they have done, and they've had reasonable follow-up. Okay? So I think this is something that in the future we might be thinking of using. Again, they've had problems with a couple of patients, ones which have failed urethroplasties and which have unhealthy vascular beds, which is the same group of patients that we also have problems with. So whether this is actually, helping us out or creating something new, I'm not sure. So they go on to say that the future needs tubular cell-loaded constructs for lung gaps, and the problem for them has been they use rabbit models, which basically they create a defect and do it. So these are normal healthy urethras. They don't have a hypospadias rabbit model, which have some abnormalities. So how much is that going to translate into actual human use is difficult to say. So to conclude, there are some increasing incidents of hypospadias. I think we can have comments from all around. Familial cases are probably more common than we think. Genetics and epigenetics of hypospadias is being now understood. And maybe in the future we might talk about treating or managing these patients before we get to it. Antenatal diagnosis is getting more and more accurate. We are understanding more about the anatomy and the biomechanical properties of the abnormal hypospadias urethra or spongiosum with different techniques. And it might help us to make a decision on what type of repair, whether we tubularize or maybe put an onlay instead of tubularizing, or we need something extra. And this is something that I think I'm quite interested in following up and seeing whether we can do some of this in our place as well. So again, assessment of these preoperatively, now there are standardized charts, and then assessing them postoperatively in a standardized manner and helping so that they can be compared across all centers, so we get numbers which are very relevant. Is probably very, very useful and something that I think all of us should look forward to. And transition care, long-term follow-up of these patients into adulthood is probably required. And maybe the very bad proximal hypospadias, maybe it should be done in more higher volume centers. And the future tissue expanders and tissue engineering may be useful. Thank you.
Just before lunch, that's a really difficult topic to tackle, and I must congratulate you because I was wondering, what are you going to say new about hypospadias? And you've given us a whole lot of things to think about, and I'm happy you found the rabbit model very useful. They produce a lot of children, yeah. So, any one question, please, because lunch is calling, and it's only the urethra that can be replaced. That's what you're saying, yeah.
I haven't talked about any other replacement at this time.
This bladder, growing urethra have all been tried. That Atala, I think this morning there was a reference put up by somebody, Atala. He was the first person to do it, to grow urothelium on various tubes, on various scaffolds, and some of them dissolvable scaffolds. So I think it's a great future because we're going to run out of buccal mucosa sometime. Thank you very much. Thank all the speakers for a great presentation. Thank you very much for your...